day, welcome to KU Padasala. Today we will look at host pathogen interaction. Basically we will study how a plant will try to evade a pathogen and how the pathogen will try to infect the plant and destroy the plant. Okay, so this is called host pathogen interaction. So first we will see what is a host and what is a pathogen. So host is any plant. It can be a monocot plant or a dicot plant. Pathogen means it can be any infective microbe. So you have bacteria, you have fungi, virus or a nematode. So any of these can be a pathogen. So disease, what is a disease in plants is it causes, disease is a condition in which it causes the dysfunction of the normal physiology of the plant, and it can be either a biotic or as well as an abiotic stress. So that induces a change in physiology, and this can be considered as the disease. Biotic means any living organism that causes this disease. Abiotic means it can be stress. So let us say high content of salts in the soil, less water, all these are abiotic stresses, and also high intensity light is also considered an abiotic stress, or increase in temperature. So these are all abiotic stress. So today we will focus only on biotic factors and that cause disease. So how does the plant interact with the pathogen? So there are different ways. So there can be plants that will evade the pathogen. So the pathogen has no role, or the plant does not interact with the plant. So this is the first mode of interaction, where there is no give and take between the pathogen as well as the host. Second one is the mutual adjustment of the plant to the pathogen. So plant will allow the pathogen to enter inside its body, and it will allow the pathogen to divide as well as replicate, but the plant does not show any symptoms, any bad symptoms. So this is the second mechanism by which the plant can accommodate a pathogen. So this is called tolerance. Okay, the third one is resistance. Resistance means the plant will prevent the pathogen from entering inside its body, and it will destroy the pathogen from moving or spreading inside its cells. So this is the third mechanism, or the antagonistic mechanism of the plant as well as the pathogen. So these are the different ways in which the pathogen as well as the plant can interact. So you can see pathogens can be of two types. You can consider them as virulent pathogens as well as avirulent pathogens. So what are virulent pathogens? Pathogens that can induce changes or cause disease symptoms in plants are known as virulent pathogens. So here from the picture you can see this is a leaf. So infections are there, in which you can see different modes of virulence. The first figure shows that the whole tissue is necrotic and it has killed the plant tissue. In the second figure you can see that the plant tissue is still intact, but it is showing some symptoms of death. Third one, you have parts of the tissues that are killed completely, some of them are surviving. So this is the different grades of avirulence that you can see. So in a avirulent pathogen, it does not cause any disease symptom to the plant. The pathogen will be present inside the body, but it will not show any symptoms of disease. So pathogens can again be classified into biotrophs, necrotrophs as well as hemibiotrophs based on the disease symptoms that it produces. So the first type of pathogens which are most dangerous to plants are known as necrotrophic pathogens or necrotrophs. What do? They will destroy the plant cells. They will kill the plant cells by inducing the production of certain toxins. So these are called necrotrophic pathogens. Second one is called the biotrophic pathogen. So here the pathogen will enter inside the plant, but it does not kill the plant cells. It will live within the plant cells, and there will be a give and take association. That is, the plant cell will accommodate the pathogen, and the pathogen will divide within the plant cell, but it will not show any disease. The plant does not show any disease symptoms. So this can be considered as a tolerance. And the third one is called a hemibiotroph. In a hemibiotrophic pathogen, initially the pathogen is a biotroph. That is, the pathogen is allowed to divide inside the living cell of the plant. But at later stages, what happens? Once the pathogen divides and produces more number of its own, it will kill the plant cell and it will destroy it, and it will live in dead tissues within the plant as a parasite. So this is the hemibiotrophic pathogen. So pathogens you can divide them into either necrotrophic, biotrophic or hemibiotropic pathogens. So in this figure you can see the examples of all the three. The first is the Botatus cineraria. So it is a necrotrophic pathogen. And second figure you can see the biotrophic pathogen. It is a Hyloperinospora arabidopsis, and third one is a hemibiotroph, that is Pseudomonas syringae. So these are the examples of each and every organism. And next is what are the steps that the plant pathogen interaction. So first step is recognition of the pathogen as non-self by the plant. So how does it do? So the pathogens will produce certain molecules. These molecules are called elicitors. These elicitors are detected by the plant cell, and that is how the plant recognizes the pathogen. So the first step is recognition. Second step is the transduction. What is that transduction referred to? Is the movement of signals from the extracellular membrane towards the nucleus, so that where the information is passed in a step-by-step manner, so that the plant will recognize and it will respond to the pathogen attack. So that is the transduction. Transduction involves different steps. The first step of transduction is called perception. So perception is a surface level phenomenon. So here the elicitor that is secreted by the pathogen is detected by a receptor on the plant cell. So there are several receptors that are present on the surface of the plant cell, and the elicitor that is secreted by the pathogen is detected by the plant cell receptor. So this is the first step. This step is called perception. Second step is called signaling. So in this signaling, from the cell surface receptor, signals are produced, and these are transmitted from the cell surface to the nucleus. So what are the different steps from which the signal is transduced to the surface of the cell, from the surface to the chloroplast, mitochondria and other organelles including the nucleus. And the third and the final step is response. So after the plant has sensed that there is a pathogen inside it, it has to respond to that. So either it will secrete certain chemicals, or it will synthesize certain molecules that will prevent the entry or that will prevent the growth of the pathogen, so that the plant cell is able to survive. So this is called the response. And finally the final step is called culmination. It will either result in the death of the plant, or it will either result in the tolerance of the plant. So the plant will either get susceptible and die itself, or it will be able to tolerate the pathogen and it will be able to survive. So this is the culmination or the final outcome of the infection. So in recognition and perception, you have an example that I have given in this figure. So this is an example of two examples are there. The first one is a pathogen, which is a powdery mildew infection. So in a powdery mildew, as you know, it is a fungal infection. So here the fungus is produced in the form of spores. These spores will come and fall on the surface of the plant cell. So once the fungal spore germinates, it will produce a structure which is called the appressorium. This appressorium will cause a foothold on the surface of the plant cell, and slowly it will enter inside the plant cell, and it will produce parasitic roots, and these parasitic roots like forms are known as haustoria. Okay, so that is the first image that you can see. So there are certain receptors on the surface of the plant cell. So first one is called the SERK1. So it is a lysine motif kinase receptor, and it is detecting the chitin in the fungal cell wall. So fungal cell wall has chitin, and this chitin is recognized by the SERK1 receptor, which is present on the surface of the cell. The second receptor is called the RAM2, which is the reduced arbuscular mycorrhization 2 gene. So what does this do is, it will detect the presence of appressorium. Appressorium is also called the hyphopodium. So the fungus will produce an appressorium or a hyphopodium, and this is detected by the receptor, that is RAM2. The third molecule that I have mentioned in this figure is the MLO locus, which is the mildew locus. It is also a surface recognition molecule, and it will infer resistance to the mildew infection. So these are some of the examples. So as you can see in the figure 2, you have the production of the hyphopodium, and the hyphopodium is entering inside the plant cell, and it is producing the arbusculation or the roots within the cortex of the roots. So this is how the entry of the pathogen takes place. So once the pathogen enters inside the plant cell will produce certain molecules which are called effector molecules, and these effectors produced by the pathogen will alter the signaling mechanism within the plant cell, and it will take the nutrients from that is produced from the plant cell for its own growth. This is the mechanism by which the plant pathogen interaction takes place. Okay. So in this we can see the transduction. I have already mentioned what is the signal transduction. So once the signal is perceived by the surface receptors, this is transduced. So the first step is the detection of non-self molecules. So molecules that are present on the fungus is never present within the plant cell. So these molecules are called non-self molecules that are detected by the plant cell. That is the first step. Second step is the movement of signaling from the surface. So the cell surface, as you can see, has a fluid mosaic membrane model. So there are certain microdomains. So these microdomains will sensitize the signaling, and they will produce certain molecules which are called secondary messengers. Cyclic AMP is an example of a secondary messenger. So these secondary messengers will go and they will bind to different receptors within the nucleus, and then they will produce the response. So what are the kinds of response? So mostly genes or transcription factors are produced, and these transcription factors in turn will induce the production of certain genes, and these genes will produce proteins, and these proteins will be involved in the synthesis of macromolecules. So the first kind of chemical molecules that are synthesized are known as monolignals. So these are nothing but secondary wall molecules, and they will increase the production of lignin or the secondary wall material, and it will prevent the entry of the pathogen inside the plant cell. And the second step is the transport of these membrane proteins through the Golgi complex or the endoplasmic reticulum to the cell surface, and finally once it reaches the cell surface, they will cross link with other phenolic molecules, and they will produce a barrier for the entry of the pathogen. So this is the step in signal transduction. So from perception to signal transduction, what are the different steps? That is what is mentioned in this cartoon. So in the defense response, basically the fungal cell wall has chitin. So chitin, if it is inducing the signaling, what it does is it will induce the production of defense responsive genes, and these genes will in turn give tolerance to the infection. So what are the different types of responses? So you can say that the plant has two types of defenses broadly: passive defense mechanisms and active defense mechanisms. Passive defense mechanisms can again be divided into two: physical barriers as well as chemical barriers. So what are the kinds of physical barriers that are passively present in the plant cell that prevents the entry of pathogen? First one is the wax. Most plants will have a waxy coating. Second one is the cuticle. Third one is the cell wall, and we have the stomata as well as the lenticels. So these are all physical barriers that prevent the entry of pathogen inside the plant cell. Coming to the chemical barriers, what are the different chemical barriers? First one is the deprivation of nutrients. Second one is pH. Third one is called phytoanticipants. So phytoanticipants are molecules that are already present within the plant cell even before the pathogen attack. These anticipants will prevent the pathogen from entering inside the plant cell. And the fourth kind of chemicals that are produced is called plant defensins. So these molecules, they are basically phytoalexins, and these are produced only after the pathogen attacks the plant cell. So they are only produced after the pathogen attack. So once the pathogen attacks the plant cell, these chemicals are synthesized. So this is the difference between phytoanticipin and phytoalexin. And then we come to active defenses. Active defenses can be two types, basically based on the time in which they are produced. So you can say rapid as well as delayed. So active defenses that are produced rapidly. First active defense is membrane functions. So membrane functions is getting altered. So there is a increased production of reactive oxygen species. You can say this is called oxidative burst. Third is the cell wall reinforcement. So I have already mentioned secondary walls materials will be synthesized, lignin, suberin, all these secondary wall materials will be produced and they will be deposited on the secondary wall. So this is the cell wall reinforcement mechanism. Third and fourth mechanism is the hypersensitive cell death. Hypersensitive response produces cell death. So what does it do? So whichever cell has the pathogen within it, these cells will die and they will prevent the pathogen from entering another healthy cell. So it is like a suicidal death, wherein the death of the infected cell will prevent the infection of the normal cells within the plant. So that is called hypersensitive cell death. And the fourth one is accumulation of phytoalexins. So I already mentioned phytoalexins are molecules or secondary metabolites that prevent the growth of the pathogen within the plant. So there will be accumulation of these phytoalexins and they will prevent the further growth of the pathogen. And coming to the delayed response, you have the production of certain proteins, and these are known as PRR proteins, pathogenesis related proteins, PRR proteins. So pathogenesis related proteins we can broadly call them, and these proteins will also prevent the pathogen growth within the plant cell. So it is a delayed response. The second one is systemic and acquired resistance. So it is also abbreviated at SAR, systemic and acquired resistance. So here also there is a delayed response, and there are a whole lot of mechanisms that will prevent the growth of the pathogen within the plant cell. Third is pathogen containment. Pathogen containment refers to the fact that pathogen is contained in a particular tissue or a particular area within the plant cell. So these are the two passive as well as active mechanisms. Passive, again we divided them into physical and chemical barriers. Active we divided into rapid as well as delayed responses. So this is the overall picture. Finally, coming to the culmination, if you this is a cartoon that shows the attack by either a biotroph or a necrotroph or a hemibiotrophic pathogen. So if the biotroph is entering inside the plant, what happens is normally the plant will induce senescence. So the plant will senesce early and it will prevent the further division and as well as the growth of the pathogen. Or what happens is that the plant will completely prevent the biotroph from growing. So that is another response. If it is a hemibiotroph, initially the plant will tolerate the hemibiotroph. At a later stage it will die of senescence. So this is the response to hemibiotroph. In a necrotroph, most likely the plant cell will induce senescence and it will prevent the further growth. So if the plant cell is susceptible to a necrotroph, it will die. So that is susceptibility. And if the plant cell is able to tolerate the pathogen, it can be a biotroph or a hemibiotroph, that is called resistance. So there is no pure resistance or there is no 100% susceptibility. Plants will fall in between these two phenomena. So what are the different responses, plant immune responses that will prevent the growth of the pathogen? The first response is called basal response. Here it produces the activation of certain genes, and these genes transcribe and produce pathogenesis associated molecular patterns, and pathogenesis associated molecular patterns are called PAMPs or PAMPs in short form. And second is the hypersensitive response. So what is hypersensitive response is the plant cells will undergo cell death, and this particular kind of cell death is called apoptosis or programmed cell death, wherein the plant cell prevents the entry of the pathogen to the next healthy cell by undergoing cell death. Third one is systemic as well as acquired resistance. Here the entire plant becomes resistant to the pathogen attack, and this is mainly the systemic and acquired resistance is mainly through a pathway, and this pathway is called salicylic acid pathway or the SA pathway. So the main molecule that is responsible for the systemic and allied resistance is salicylic acid, systemic as well as acquired resistance. And third is the induced systemic resistance or ISR, and induced systemic resistance is produced mainly through the jasmonic acid pathway or the JA pathway, and or it can be either through jasmonic acid pathway or ethylene. Both of them are involved in the induced systemic resistance or the ISR pathway, and mainly it is active against herbivory, that is wherein herbivores will attack the plant and destroy it. And the final one is the non-host immunity. So we come to the basal response. So here basal response is mediated by pattern recognition receptors or PRR proteins, and they are triggered by certain molecules or elicitors that are produced by the pathogen. So mainly these molecules can be either chitin on the fungal cell wall, chitosan, or flagellin, the molecule that is present in the flagella of bacteria. So any of these can be acting as the pattern recognition receptors or PRRs. These will be sensed by the receptors in the plant cell, and they will produce certain responses. So what are the different types of responses? The cell wall strengthening will occur. The plant cell will produce callose plugs. and the plant cells can also synthesize phenolic compounds, and finally it will produce lignin secretion. So these together can be called as pathogen triggered immunity. Why PTI, or pathogen triggered immunity? Because this immunity is triggered during the pathogen attack. That is why it is called pathogen triggered immunity. So from this cartoon you can see it very clearly. So once the pathogen is present and the chitin or the molecule that is present on the surface of the pathogen is sensed by the plant, it will induce a production of reactive oxygen species, and this is called the pathogen triggered immunity. So pathogen triggered immunity can be inactivated. So basically this is a phosphorylation reaction. So there are certain kinases that are produced, and these are producing the pathogen triggered immunity, and in turn if you have phosphatases that prevent this, the phosphatases will dephosphorylate the kinase enzymes, and finally the pathogen triggered immunity is come to a lower level. So that is called inactivation. So you can either activate or inactivate the pathogen triggered immunity. So how does the plant prevent the vascular spread? So you know, plant has different conducting tissues, xylem, phloem, etc. are there. These conducting tissues can be transporting the pathogen throughout the body of the plant. So there are certain mechanisms that prevent the movement of pathogens through these conducting channels, and you can see that in the image. So here you can see the cross section of a xylem vessel, and once the fungus, here I have shown the example of a fungal pathogen. So Aprosoria and hyphae are entering inside the xylem vessel, and it is trying to move from one cell to the other. So what happens is that these cells will produce certain invaginations of the wall, and these are known as tyloses. So these tyloses will block the vessels, and it will prevent the further spread of the pathogen. So this is one of the mechanisms. And I was talking about hypersensitive response. So hypersensitive response can be induced by chemicals also. So hypersensitive response normally produces programmed cell death or apoptosis, and it will prevent the movement of pathogen further inside the plant. And hypersensitive response is shown in this figure, and here the sequence of events are shown. So here the Aprosorium of the fungus is entering inside the plant cell, and the plant cell is turning senescent, and it is slowly dying, and it is preventing the movement of the Aprosorium or the hyphae to the neighboring cells, and the neighboring cells that are present next to the dying cell also produces certain lignification factors that will prevent the entry of the pathogen into them, the healthy cells. So this is a cartoon that shows the sequence of events in a hypersensitive response against mainly a fungal attack, So here you can see the Pseudomonas syringae attack on a Arabidopsis plant. Here also it is present. So the genes inside the cells are responsible for the susceptible response or the resistance response. So in the presence of certain suppressing factors, the plant will get susceptible to the infection by the Pseudomonas syringae, whereas in resistant plants the pathogen will induce hypersensitive cell death, and it will prevent the further growth of the plant. So this is based on the presence or absence of certain proteins. Here it is the RPM1 protein, and the presence of RPM1 protein will induce resistance to Pseudomonas syringae, whereas the absence of that protein will induce the cell death. So this is the response by which the plants will induce either susceptibility response or resistance response. So here we have another mechanism which is called effector triggered immunity or ETI. So here the pathogens produce certain genes, and these genes induce the resistance genes that are already present. So lot of resistance genes will be pooled together, and this is called a qualitative resistance. So this qualitative resistance is very specific to a particular pathogen. It is not common. So this qualitative resistance will prevent the pathogen. Another mechanism is that of combined immunity, that is the plant resistant mechanisms will be pooled together, and this kind of a response is known as quantitative resistance. So there are different mechanisms, all these pooled together will induce a response. So there is a relative accumulation of these defense related proteins, and then finally they resist in the response. So here in this figure you can see the necrotroph as well as the biotroph how they induce the systemic and acquired resistance, and how resistance proteins are involved, and how it is inducing the pathogen triggered immunity or the effector triggered immunity. So, what is the difference between these two in the case of a biotroph as well as a necrotroph, and how hypersensitive response prevents the pathogen from further attacking other cells. So, all these together will either result in susceptibility to necrotrophs or resistance to biotrophs. That is what happens. So, I have already explained the salicylic acid mechanism. So, salicylic acid pathway has an advantage. So, here plants are incapable of moving from one place to the other. So, what do the plants do? So, once a particular plant in a field is getting infected, the plants will produce a volatile chemical, and in this case the example given is methyl salicylate. So, methyl salicylate is an isoform of salicylic acid. Methyl salicylate is volatile. So, once a plant is infected by a pathogen, that particular plant will produce methyl salicylate, and this methyl salicylate, which is volatile, will sensitize other plants which are very close to this particular plant, and then it will induce a response. That is, it will prevent the pathogen from infecting other healthy plants in that particular field. So, this is another mechanism by which plants will transmit the resistance to other plants that is next to them. So, salicylic acid pathway, as I mentioned, is mainly for defense against biotrophs, whereas jasmonic acid pathway is basically preventing the attack of necrotrophic pathogens, and these together have crosstalk. So, salicylic acid pathway as well as jasmonic acid pathway, they communicate with each other and they will result in defense response in a plant. So, this is the systemic and acquired resistance and induced systemic resistance in a gist. So, this will result in resistance against biotrophic pathogens as well as resistance against necrotrophic pathogens by the interplay between salicylic acid pathway as well as the jasmonic acid pathway. So, this is a cartoon that shows the release of a hormone. So, this hormone is a polyketide hormone which is called histamine, and this hormone, once there is a herbivory, once there is a tissue damage, this will be secreted and this will be produced inside the phloem. So, once this is secreted inside the phloem, it will be received by the receptors on the cell surface of other cells, and they will produce lipase. This lipase enzyme, what does it do? It will destroy the membrane lipids. So, membrane lipids, once they are digested, they will produce free linolenic acid, and these free linolenic acid molecules will trigger the jasmonic acid pathway, and they will result in the activation of protein inhibitory enzymes, and finally the pathogen or herbivory response will take place within the plant. So, this is the if the pathogen has certain elicitor molecules that are present outside on its cell surface, they are against activated by genes that are present in the plant. These are resistance genes, and these resistance genes will induce the production of hypersensitive response or phytoalexin synthesis, lignin biosynthesis, or salicylic acid synthesis. Salicylic acid synthesis will result in systemic acquired resistance, whereas hypersensitivity response will induce the systemic resistance. So, either way, the whole mechanism will prevent the entry of pathogen or the it will prevent the destruction of pathogen attack. So, these are the regulatory nodes. You have ethylene pathway, you have jasmonic acid pathway, you have salicylic acid pathway, you have abscisic acid pathway, and you have the reactive oxygen species pathway. All these pathways will interfere, and finally they will either produce systemic resistance or induce systemic resistance, and finally result in either resistance to the pathogen or susceptibility to the pathogen once the pathogen attacks. So, these are some of the examples of phytoalexins. So, I already mentioned phytoalexins are produced only under pathogen attack. So, only under pathogen entry phytoalexins are produced. So, inorganic phytoalexins example is sulfur, and basically it is produced in cocoa plants. Phenolic phytoalexins examples are chlorogenic acid as aveloamines, mostly it is produced in potato, tobacco, apple, and some cereals. Terpenoids include capsidiol, rishetin, hypomenaramine, and gossypol. They are basically produced in tobacco, tomato, sweet potato, and cotton. And the main pathway, which is the phenylpropanoid pathway, examples are visatin, fasciotin, glyceolins, and medicarpin. These are produced by alfalfa, clover, broad bean, chickpea, etc. Acetinic molecules include saffenol and viarone. They are produced in broad bean and safflower. Still bean pathways is also there. Resveratrol is an example of a still bean, as well as patatacins. They are produced in yams as well as in grapefruit. And you have indole sulfur molecules, that is camelexin as well as brassilins. Basically they are produced in Arabidopsis, cabbage, rape and turnip. So these are the different types of phytoalexins. And if you see at the timeline of resistance, so responses can be taking place in minutes, hours and days. So in terms of time, if you divide the responses, you can say that first response which happens in minutes is the membrane depolarization. So cell membrane depolarization will take place and electrolyte leakage occurs. So the first step is the production of reactive oxygen species or ROS generation. This ROS generation is followed by the expression of genes involved in phytoalexin synthesis. So all these three mechanisms will take place within minutes of pathogen entry or within minutes of tissue damage. Okay. That is the response that happens in minutes. So what are the responses that takes place in hours? So in time, let us say after 1 hour of infection, there is a sudden burst of reactive oxygen species, and this is called oxidative burst. Second step is membrane peroxidation. So membrane lipids will get oxidized, and this is the second step. Third step is rise in salicylic acid levels, and then there will be cytoplasmic aggregation of membranes, as well as the cells will collapse and it will result in the hypersensitive cell death. So this will happen hours after the infection, and it is followed by cell wall reinforcing mechanisms, that is the synthesis of lignins, secondary cell wall material. All this will happen in hours. Then what is the response that is happening in days? That is more delayed response. In days, we have the accumulation of pathogenesis related proteins, and it will induce what is called as systemic and acquired resistance. So this is how the plant, these are the different mechanisms by which the plant will prevent the entry of pathogen or it tolerates. So in order to study this, I have given an example here. So we have the black pepper, which is the Piper nigrum, and the Phytophthora capsaicei, which is a fungus that attacks the black pepper. So black pepper, it is an important cash crop for Kerala, and here we understand the mechanism as to how the black pepper infection takes place by Phytophthora and how we can prevent it. So we collected the plant material, and we also collected the Phytophthora from the Agriculture College Vellani, and we cultured them in the potato dicrose agar medium. And what we did is we stimulated the response. The defense genes were stimulated by the addition of chitin. So here we have used chitin as well as chitosan as the elicitor molecules, and these elicitor molecules were provided in different ways. We either used chitin in the roots directly, or we used to spray it on the leaves. So chitin as well as chitosan were applied together or different. So this list provides the different kinds of treatments that we did for the plant, and how the pathogen was infected. So we made small cuts into the plant, and these pathogens were infected by growing them in a potato dextrose agar and then putting this PDA discs on top of the injured plant part, and then we covered with cotton, moist cotton, so that the pathogen does not dry. So here you can see after 15 days of pathogen inoculation, you can see the plants have wilted and died. So this is classical susceptibility response. These plants were not treated with the elicitor. So here you can see another treatment group. Here you can see after 15 days of pathogen treatment also these elicitor treated plants are surviving, and they are comparatively healthy, as similar to the healthy plants, normal plants that were uninfected. So here you can see that treatment with chitosan or chitin is helping the plant to induce the defense response, and this is also true in leaves. We have done leaf assays. So in the leaf assay, we take the leaf out, we keep it in a petri dish, and we inoculate the pathogen. Here also chitin as well as chitosan treatment prevented the movement of pathogen to a certain level in the pathogen treated ones. So we also looked at the sections, anatomical sections. So as I already mentioned, wall formation, secondary wall formation is a major mechanism by which the plant will prevent the pathogen entry. Here you can see from this picture in the elicited treated group, you can see the secondary wall formation is much higher. So, you can see the secondary wall formation is increased in the treated group, whereas in the control group you do not see the secondary wall formation. So, in order to see if this is true, we did the quantitative PCR analysis, and we looked at some genes that are involved in secondary wall synthesis. We looked at the PAL gene, that is phenyl ammonia lyase. So, here you can see that the plants which were pre-treated with chitin, they showed a reduced expression of the PAL gene, whereas C4H hydroxylase or cinnamate 4 hydroxylase gene was upregulated, and this upregulation was 5 times compared to the normal plants. So, here you can see that there is an increased fold expression of C4H gene, and this is because of the elevation of the phenyl propionate pathway in this particular. Another example that I have given here is of ginger. Another type of pathogen is the Pythium. So, Pythium is a biotrophic pathogen that I have already mentioned. So, here we have used ginger as a host plant. Here you can see infection with Pythium will kill the plant from these pictures. So, again we have given elicited treatment with chitin or chitosan, and again our elicited treatment led to the tolerance of the infection by these plants. So, here you can see in cotton blue staining, the Pythium hyphae are seen within the host cell, okay, and once you give the elicited treatment, there is the wall formation or the secondary wall formation takes place, and this is the picture that shows the secondary walls. So, we stain the wall material with fluoroglucinol. Fluoroglucinol is a specific stain that that stains lignin. So, here you can see there is a increased secondary wall formation, and it prevents the entry of the pathogen within the plant. Again, we also looked at the expression of genes here using qPCR. The genes we analyzed was PAL, phenyl ammonia lyase, and catalase. The catalase gene was increased around 3.9 times or 4 times roughly, whereas you can see that there is an increased production of the phenyl ammonia lyase gene, that is 10 times that particular gene was activated by infection. So, here what I wanted to mention is that by elicited treatment we can prime the defense response to plants to a particular pathogen. So, elicitors will prevent the entry of pathogens, or it can either tolerate the plants will be so acclimatized by elicited treatment that they will be able to tolerate the infection, and you can prevent the destruction of the whole crop. That is what. Thank you.